This next lecture is going to be a brief review of biochemistry and some of the chemistry concepts that you should have gotten in other courses before taking this course. So first of all, just to review some basic chemistry terms, matter is a term that is commonly used in chemistry. Basically anything that occupies space and has mass. And mass is how much matter is within an object. So for the human body, we commonly talk about mass and weight as being the same, but weight is actually a slightly different term, and weight actually refers to the effect of gravity acting on a body's mass. All right, and then some terms that you should remember from chemistry: element and atom. So element, you've all seen the periodic table of elements, and here it is right here. And basically an element is just defined as the simplest type of matter with unique properties. And then an atom is the smallest particle of an element that retains the chemical properties of that element. And so you may remember what an atom looks like, an atomic structure, which I'm going to show you on the next slide, but an atom has a nucleus and then these shells going around it. All right, so atomic structure, this is an atom, and if you look at the atomic structure, we have a nucleus, as I was saying, and within the nucleus you have positively charged substances called protons, and also neutral, which means it's not positive or negative, substances in there called neutrons. So protons and neutrons are found in the nucleus. So protons positive, neutrons are neutral. Then going around the nucleus in these what are called energy shells are electrons, which have a negative charge. So then each of the atoms that you may come across, and again, this is not a chemistry class, but they all have different numbers of protons, and that gives each atom its atomic number. So some of the atoms that we're going to come across a lot in this course are going to be like hydrogen, carbon, oxygen, so ones that you've seen in a basic chemistry course. So atoms can bind together. This is really important in anatomy and physiology, but there are three different types of bonds that I know you've seen in other courses. So we'll talk about ionic bonds first. So ionic bonds are relatively simple, and it's basically if you get a positive and negative charge attracted to one another. So if an atom gains or loses an electron, it becomes an ion. So remember, electrons are negative. So if an atom loses an electron, it becomes slightly positive, has a slightly positive charge. If it gains an electron, it will get a slightly negative charge. So an easy example of this is sodium and chloride. So the way sodium commonly exists, it's as an ion. So an ion is just referring to an atom that has a charge on it, positive or negative. And so sodium commonly loses an electron, so it has a positive charge. So we commonly write sodium like this. Na is the abbreviation for sodium, has a positive charge. Chloride commonly gains an electron, so we write it like this, Cl negative. And those are charged particles, which are known as ions. So we could call this a sodium ion, we could call this a chloride ion. The names that we give these ions if they have a positive or negative charge is cation if it's positive, anion if it's negative. So sodium would be a cation, chloride would be an anion. And getting back to our bonds, these opposite charges attract one another. And so we would end up with sodium chloride. That positive of sodium attracts the negative of chloride, and they get together and form a molecule. So that's the basis for an ionic bond. Positive and negative attracted to one another. Other bonds, there's covalent. Covalent, the key thing is sharing of electrons. So the atoms share electrons. It could be single pair or double pair. So our single bond would be one pair of electrons are shared, double bond would be two pairs of electrons are shared. And a few examples up here. So this methane molecule, so you have carbon and it has 4 hydrogens, and you can see that the carbon and each hydrogen are sharing one pair of electrons. So that would be a single bond. If you look at carbon dioxide, you got carbon and 2 oxygens. You can see in this case they're sharing 2 pairs. And so these are both examples of covalent bonds because the atoms are sharing electrons. And then just to add another layer to this, you can have what are known as polar or nonpolar covalent bonds. So in the examples up here, we have pretty much equal sharing but of the atoms or the electrons between these atoms. You see the same thing here. So that would be nonpolar, a nonpolar covalent bond, so there's equal sharing. In the instance of water, where you have 2 hydrogens and oxygen, so H2O, you can see they're not equally sharing these electrons. So oxygen is pulling the electrons closer to it. And so that unequal sharing makes this what we call a polar covalent bond. And so what ends up happening, since the electrons are closer to the oxygen, it makes that oxygen tend to be a little bit more negative and the hydrogens tend to be a little more positive because the electrons are closer to the oxygen. And this is one of the reason that water is considered a polar molecule. All right, and then lastly, hydrogen bonds. So this is important when we talk about DNA. So hydrogen bonds, if you've seen the structure of DNA, which I'm sure you have, the 2 strands of DNA are held together by hydrogen bonds. And so hydrogen bonds, by definition, are really weak. And basically they consist of a bond between a hydrogen and an electronegative atom. All right, so hydrogen, let's take 2 water molecules in this case. So you have a hydrogen here, this is a water molecule, 2 H's, so 2 hydrogens and oxygen, H2O. And so this negative, slightly negative charge on oxygen is being pulled towards this hydrogen, which is slightly positive, and that makes a hydrogen bond. So again, hydrogen bonds are really important when you look at nucleic acids such as DNA. All right, so shifting gears a little bit, I wanted to bring up the concept of acids and bases. So by definition, an acid is a proton donor. So a proton is basically a hydrogen ion, so H+. So if you have an acid, it will release those protons when it's dissolved in water. So this is hydrochloric acid, very strong acid. If it dissociates, it's going to release a hydrogen, and the chloride ion is released also. So you can think of this as donating, this HCl is donating the proton. Bases, on the other hand, are proton acceptors. So this NaOH is, if we have it where it can react with a proton, it's going to accept it, and it's going to basically dissociate into sodium and water. So if you look at all the molecules that are accounted for here, so H2O and then sodium itself. So these are proton acceptors. So this this molecule would accept these hydrogens or protons, and you would get this dissociation like this. Another example is ammonia. So if we have NH3, it will accept a proton, and you end up with NH4. So proton acceptors are what bases is a good way to define what a base is. All right, so whether something is acidic or basic is described in the pH scale. And you've seen the pH scale before, 0 to 14. 7 is neutral, meaning it's not acidic or it's not basic at all. And then as you go lower towards 0, these are all acids, and the strongest acid would be 0. And so 6 would be, for example, a relatively mild acid, pretty weak acid. Same thing with the bases. So you have neutral as you go from 7 to 14. The higher you get, the stronger the base. So you can see like weak bases such as baking soda, all the way to stronger bases like ammonia. So pH is very relevant in the human body because within the human body we do have what's considered an internal pH that's slightly basic, and most of that is a result of the blood. So the blood has a pH range of 7.35 to 7.45. But I did want to point out that depending on the tissue or organ of the body, the pH can change. So if you look at like the pH of saliva, it has a slightly different range. It can become more acidic. The stomach is a very unique example because it has a really, really low pH, meaning it's very acidic. So it ranges from 1.5 to 4, and that's due to a lot of hydrochloric acid in the stomach. All right, some terms that you should be familiar with: inorganic versus organic. Inorganic, by definition, means it doesn't have carbon, but there is some exceptions to this, obviously, as you're going to see below. Organic means it contains carbon-containing molecules. So, exceptions: carbon dioxide is considered an inorganic molecule. Carbon monoxide, same thing. Even though it has carbon, carbon dioxide is considered an inorganic molecule. But for the most part, the molecules with carbon are going to be classified as organic. And we're going to go through several of those in a few minutes. So inorganic molecules that are important in physiology include oxygen. We need that for metabolism. We get it from the atmosphere. Carbon dioxide is a byproduct of metabolism. We release it via respiration after we utilize oxygen for energy transfer. Water is really important. It makes up about 60 to 80% of the volume of most cells, and is a huge component of the human body. So it's very important for temperature regulation, protection, transportation, serves as a medium for chemical reactions. And switching now to the organic molecules that we're going to spend some time on this semester. So we have carbohydrates, lipids, also known as fats. We have proteins, and we have nucleic acids, such as DNA. So I want to run through a pretty brief overview of these organic molecules that you'll see throughout the semester. And this is going to be important also when we talk about like the cell membrane, because the cell membrane is going to have a lot of lipids, a lot of proteins, even some carbohydrates. Then we talk about DNA, that's actually a nucleic acid. So brief overview of carbohydrates, made up of carbon, hydrogen, oxygen. They exist in either as monosaccharides, disaccharides, polysaccharides, and that just refers to the number of units. So I put some examples here, but really what you're going to see probably the most of this class, like monosaccharides, the big one we're going to come across is glucose, which is your blood sugar. Polysaccharides, glycogen, which is how we store carbohydrate in the body, and starch, which is a plant form of a long chain of monosaccharides. So let me show you a picture, and I think this will make a little more sense. So if you look at monosaccharides, if you look at the chemical structure, there's basically one unit. So you have glucose or fructose. Both of these are monosaccharides, but the key point is it's one unit. Glucose, remember, is our blood sugar. Disaccharide would be 2 units. So here's an example, one called maltose. 2 units linked together, that's a disaccharide. Another good example is sucrose, which is table sugar. Polysaccharide, on the other hand, is 3 or more. So if you have a lot of these monosaccharides linked together, that is a polysaccharide. And as I was alluding to on the previous slide, the 2 big examples of polysaccharides that are important in physiology are glycogen, which is how we store carbohydrate in the body. We store it in the liver and also within muscle. And then starch is a plant form of polysaccharide. So if you're eating a food like potatoes or pasta, that has a lot of starch, and all starch is a long chain of glucose molecules. All right, so looking in more structures, here's the structure of glucose, which is a monosaccharide, and here's glycogen. Each one of these little round circles is a glucose molecule, and so like I was saying, glycogen is just a long chain of glucose. This is how we store carbohydrate in the body. All right, moving on to fats or lipids, also made of carbon, hydrogen, and oxygen, different forms. What we're probably going to see the most of in here, we'll see a little bit of fatty acids, triglycerides. When we talk about the cell membrane, lots of phospholipids, and then steroids also. Steroids have a variety of roles in the human body, including key components of the cell membrane, but also they serve as hormones. So fatty acids look like this. So fatty acids are a long chain of carbons with hydrogens attached. And these are really important because we can get a lot of energy out of these. So these can produce a lot of energy. We can transfer a lot of energy out of these. Looking more detailed at the fatty acids, so if there's no double bonds between the carbons, we call it a saturated fatty acid. And let me show you this really quick. So in this example, these are all single bonds between these carbons. So that makes this a saturated fatty acid. If there was a double bond in here, or more than one double bond, it would be unsaturated. And so that's the difference between saturated and unsaturated. So saturated, no double bonds between the carbons. Monounsaturated means there's one. Polyunsaturated means there's more than one. So some quick examples here. This is saturated. If you look closely, these are all single bonds between the carbons. You look here, there's a couple of, looks like 3 double bonds, one here, one here, one here. So this would be polyunsaturated. And then if we look at this example right here, there's only 1 double bond, so that would be technically monounsaturated. And you see another saturated one right here. All right, so triglycerides contain actually these fatty acids, so it always makes sense to talk about the fatty acids first. So a triglyceride is a molecule of glycerol, which is this 3-carbon unit right here, with 3 fatty acids attached. And those fatty acids could be saturated or unsaturated. These are really important, these triglycerides, because they can be used for energy because they're loaded with fatty acids. And also, this is how we store fat in the body. So if you look at fat cells, which are called adipocytes, they're loaded with these triglycerides. Even muscle has a lot of triglycerides because we can get so much energy. So when your muscle needs energy, it can get it directly from those triglycerides found within the muscle itself. So this is the major storage form of fat in adipose and a lot in skeletal muscle also. And that's also the major form that we get fat in the diet. So kind of an archaic picture, but these are supposed to represent 3 fatty acids. You have glycerol, and then they get together and form a triglyceride. Not the best picture, but I think it gets the point across. All right, so phospholipids. So consist of a glycerol molecule with a phosphate and 2 fatty acids. So 2 fatty acids are key. So it's a little bit different than a triglyceride. So we don't generally use these for energy transfer just because they provide more of a structural aspect in the human body. They make up the cell membranes of cells, so the barrier of cells, which we'll get to. So that's why phospholipids are so important structurally. They basically make up the membranes of the cells of the body. They also make up the membrane going around the nuclei of cells in the body. And then they also help form something called the myelin sheath, which is an insulative layer around neurons. So phospholipid will look like this. You got that head region here, and then you got the tail region here. And so the tails are the 2 fatty acids. And then you have that glycerol and phosphate molecule here. We'll talk more about this when we talk about the cell membrane. All right, and the last classification of lipids that we're going to talk about are steroids. So steroids all have this four-ring structure like this, and they have a variety of roles, not really for energy transfer, but they have roles including cholesterol. Cholesterol is a steroid. Cholesterol gets a bad rap, but it is important in the integrity of the cell membrane. And also hormones. So there's several steroid hormones like testosterone, estrogen, aldosterone, cortisol. Those are all hormones that are classified as steroids. They all have this similar structure. All right, moving on now to proteins. So proteins are also made up of carbon, hydrogen, and oxygen, but then they also have nitrogen. So proteins are made up of smaller units called amino acids, and there's about 20 naturally occurring amino acids. And I want to show you the structure of an amino acid just to refresh your memory. So all of these amino acids are going to have the exact same structure except for what's known as their side chain or R group. So this is the chemical structure of an amino acid. So there's a central carbon, there's what's called a carboxyl group on one side, an amino group which has the nitrogen on the other side, and then there's a hydrogen. And then they're going to have this side chain, which again is sometimes called the R group. This is what makes each amino acid different. So all amino acids are going to have all of these structures being exactly the same. What makes them different is this side chain. So here's another picture. Carbon, central carbon, you got a hydrogen on one side, a nitrogen containing amino group on one side, and the carboxyl group, which is the carbon and oxygen group on the other, and then the side chain, which is what makes each amino acid different. So proteins have a lot of functions in the body, but one of their key functions is just structure. So a lot of our structures in the body are composed of primarily protein. And we can transfer energy out of amino acids, but it's not the preferred mechanism for energy transfer in the human body. So just to compare it to carbohydrates or fat, which the body likes to rely on for energy, about 5 to maybe 15% of all our energy transfer is due to protein metabolism. So proteins serve more of a structural role, and they also serve as something called enzymes. So we'll get to that in a moment. But the way that these amino acids link together to form proteins, they link together via what are called peptide bonds. So amino acids link together to form proteins via these peptide bonds. So you can see there's multiple amino acids here, and they're linked together by these peptide bonds, which are denoted in green. So technically, if you want to talk about specific terminology, if you get 2 amino acids together, it's called a dipeptide. You get 3, it's called a tripeptide. It's probably just easier just to think of them as proteins, and that's fine for the purposes of this class. And then some proteins act as enzymes, which, if you remember what enzymes do, they catalyze or speed up chemical reactions. So you probably were exposed to this in a chemistry course, but an enzyme is a protein that speeds up chemical reactions, and the way it does it, it lowers what's called the activation energy, which is the energy required to start a reaction. So I like to look at this graph right here, and it's kind of showing what we would know as an exergonic reaction that overall releases energy. So if you start with what are called the reactants, you end up with what are called the products. You can see that over the course of this chemical reaction, energy is given off. So we know that as an exergonic reaction. But the important thing here is that you can see that for this reaction to proceed, you have to invest a little bit of energy to get it started. That energy invested to get it started is known as the activation energy. And what an enzyme does is it lowers that activation energy. So this green line represents the reaction without an enzyme. You can see it's going to take a lot of activation energy to get it started. Thus, it's going to take longer to get that reaction started. But an enzyme lowers that activation energy, and basically allows that reaction to proceed a lot more rapidly. And another key point about enzymes, they don't alter the amount of energy that's transferred. They just speed up the chemical reaction. And most enzymes are going to end with the suffix ASE. So if you see a word in your anatomy or physiology textbook that ends in ASE, it's likely an enzyme. All right, so a long drawn out description of how these enzymes work. This is probably a good schematic to just envision. So an enzyme is going to act on a substrate, and so for instance, in this case, the enzyme is maltase. So it acts on this substrate or reactant in this case called maltose. And the important thing to see here is that they fit together like a lock and key. And that makes an enzyme specific for a given substrate. So this enzyme won't really work on any other substrate other than the ones that it's specific for. And so these active sites that they have fit together like a lock and key. And when they get together and they match, you get what's called an enzyme substrate complex. That's where the activation energy is lowered and the reaction proceeds, and then the enzyme is released, and it's still the same, it hasn't changed at all, but the reaction proceeded. So in this case we end up with what are called products, and they're not the same as the substrate. So the substrate was reacted and formed two products. The enzyme catalyzed that reaction and was released in an unaltered form. So it's free to catalyze another reaction. So the simple way to look at this, enzyme meets a substrate, you get an enzyme substrate complex, and what's left at the end are the product and the unaltered enzyme. All right, last thing about enzymes. Enzymes are heavily influenced by temperature and pH. This is very important in the human body. So each enzyme is going to have an optimal temperature, and that's the temperature where it works the best. If the temperature gets too high, the enzyme doesn't work as well. And if the temperature gets too low, it doesn't work as well. So this is one of the reasons that it's imperative that the human body stay at an ideal temperature. Same with pH. So each enzyme works at an optimal pH. Depending on the enzyme in the body, they have different optimal pHs. But you can see these are two different enzymes. They each have an optimal pH. But if that pH drops a little bit, the enzyme doesn't work as well. If the pH goes up too much, the enzyme doesn't work as well. So you need to have an optimal pH for enzymes to function adequately. All right, the last organic molecule I want to talk about is nucleic acids. So nucleic acids are made of carbon, hydrogen, oxygen, nitrogen, and also phosphorus. So nucleic acids are long strands. They're made up of building blocks called nucleotides. And so this looks like DNA here. So you have two strands in this case. So each of these strands would represent a nucleic acid. Each one of these building blocks that has the sugar would be the nucleotide. And so looking in detail at a nucleotide, each nucleotide or building block is going to have a 5-carbon sugar. And commonly that's either going to be ribose or deoxyribose if you're talking about DNA or RNA. It'll have a nitrogenous base, and there's 4 options there we'll get to, or 5 if you're talking about RNA also. For DNA there's 4, I should say. And then there's a phosphate group. So that's a nucleotide. So focusing in on DNA, because it's such a common molecule that we need to know about in physiology, the sugar that makes up the nucleotides is going to be deoxyribose. And it's not shown well here, but it's labeled right here. So this is a nucleotide. Deoxyribose is the sugar. The 4 possible bases are adenine, thymine, cytosine, guanine. And so each one of those nucleotides is going to have one of those. And then DNA itself has 2 strands. So these 2 strands are basically nucleic acids that are made up of the nucleotides we just talked about. And they're held together by hydrogen bonds. And remember what we learned about hydrogen bonds, relatively weak, so they can actually come apart when necessary. And the bases actually link together in a very specific fashion, so that adenine on one strand will always bind to the thymine on the other strand, and the cytosine on one strand will always bind to the guanine on the other strand. And that's just always how it's going to be with DNA. So A binds to T, or T binds to A, C binds to G, or G to C. Then also just a little bit more about DNA, the way it's packaged in the cell. It wraps DNA itself, which is has a double helix model. So it kind of looks like a spiral staircase made up of these two strands. It wraps around these proteins called histones, like this that you're seeing here. And when it does that, wraps around 8 of them, and that's called a nucleosome. Then you get a long chain of that, that's called a chromatin fiber. And then it coils up a little bit, and then what the image that you're probably most familiar with is this. Kind of looks like the letter X. So that's an organized chromosome. So just before cell division, it's going to look like this, and it's actually 2 what are called sister chromatids held together. And so that is an organized chromosome, and it'll look more like this right just before cell division. So human cells, talking about what we call somatic cells, are contain 46 chromosomes, and they're organized as 23 pairs. Then our sex cells, or gametes, like the sperm and the egg, only have 23. They're not paired up. So we'll see this when we talk about the cytology lecture also. All right, RNA, or ribonucleic acid, just another nucleic acid. So very important in protein synthesis, but in this case, it's different. It's not DNA. It's not deoxyribose as a sugar. It's ribose. And it's only one strand, but you still get the nucleotides. And then there is a difference. The bases are different. Uracil replaces thymine. Other than that, it's the same. The guanine always would bind to any cytosine. And adenine, if it had to bind to something, would bind to uracil. So hopefully you've learned about the process of protein synthesis in the basic bio courses. And this, we're not going to go through the protein synthesis whole cascade in this course, but that's where you've probably seen RNA before. And as you know, there's different types. There's messenger RNA, there's transfer RNA. All right, what I'm going to finish up with here is ATP, which is really important in human physiology. It's our energy currency within the cells. It's basically the usable form of energy within the human body. All right, so ATP, if you look at its structure, it consists of a molecule called adenosine, which is adenine and the sugar ribose, and then it has 3 phosphate groups, thus the term adenosine triphosphate. So this is what it looks like. So you have the adenosine molecule, which consists of the sugar ribose and the base adenine. Then you have 1, 2, 3 phosphate groups. This energy molecule, it's the usable form of energy, and because it can store a lot of energy, and most of the energy is stored between these bonds of the phosphate. So what's going to happen is when your body needs energy, like let's say a muscle cell needs energy to contract, a phosphate will be removed. And when that phosphate is removed, the energy is released, and that energy can fuel biologic work, such as muscle contraction or some type of transportation in the body. So that's why we call ATP the energy currency. An analogy that I commonly use is you can have all of the energy in the world in the form of like carbohydrates or fats, but it's not going to be useful to the human body unless you are able to put it into that energy into the form of ATP. And there's reactions that do that. And that gives us our overall metabolism. So another way to think about it is if you relate this to money, you could have a lot of money in a different currency. Let's say it was you're in a different country that did not accept U.S. dollars. You could have all the US dollars in the world, but it's going to be useless unless you can convert it to the correct currency. So energy is very similar. You have to convert that energy, such as in carbohydrates and fats, and you have to put it into the usable form, which is ATP. All right, so energy is stored within the phosphate bonds. Energy is released when the bonds are broken. So if one phosphate is removed, you're left with what's called adenosine diphosphate because there's only two left. If you remove two, you could end up with adenosine monophosphate. But the most common reaction is this. So one phosphate will be removed, you get the energy, and you're left with ADP and a phosphate. And here's a picture showing that reaction. ATP going through a reaction, ending up with ADP and phosphate. Here's an enzyme catalyzing that reaction. But the big thing is energy is being released. That energy can help fuel biologic work.